Guess I'm here to give you my 3-year update. I think I spoke here 6 years ago, and I promised we would transition away from arrays. We would get into nano and pico genomics, and we would get heavy into computer science. Glad to report we've accomplished all of the following. So what I'm going to talk about today is really things that have come out of the core. And this is Martin's slide. I put this here. Just to remind everybody that the laboratory for, how does this work? Yeah, here we go. The laboratory for advanced genome analysis sits over here in the pipeline. And the Prostate Center is actually world-renowned for its core-based science that accelerates discovery, translation, and commercialization. Everywhere I go, people ask me, how do you guys get so much done? And so this is the sort of the technologies we have at in Laga, and so we've got still have microarray equipment. We've got lots of sequencing equipment, although small-scale sequencing. Just had a CFI grant funded that's going to bring in more sequencing capabilities, and BGI is likely to drop in a couple of their new sequencers that sequence faster and cheaper, and the claim is with slightly better quality, and they don't need any human intervention. They're fully. It's coming. It's coming. It's fully. These things are fully automated, so that the trained personnel work downstream of the sequencers, not upstream. So, we offer computer science, what I call innovation through algorithms, and this Cenk Salanap, who's a professor at Simon Fraser, and Dr. Hack, who's sitting here, and hopefully will be joining us soon, because I'm not a computer scientist, so we need all the help. I need all the help I can get. And so we co-mentor students, so they learn both biology, medicine, and CS. And so we develop tools, and so our technologies include DNA, RNA, cell-free DNA, and protein. So we use constant R&D to drive innovation and support biological and clinical research. We can work with fresh frozen, FFPE or plasma. So a lot of people think we can't work with FFPE. So I've been using it for sequencing and array CGH since 2004. And now we just published a paper showing it works fine for mass spec, despite all the rumors to the contrary. And we have many corporate partners, and we just had a large CFI grant funded. So, it looks impressive, but in terms of genome facilities, it's actually pretty small. But because of a lot of guanxi in China, we have access to the world's largest genome institute, the Beijing Genome Institute. So on the left, that was me recently being given a position at BGI in their think tank. This actually helps form policy at BGI. There's 5 Westerners invited to that position. And then just a week ago, here in Vancouver, at the Convention Center, signing an MOU with BGI. And the MOU, I won't go into all the details. And so this just shows what BGI is. You get some sense of scale. On the right, that's their new GenBank. It's a $1 billion U.S. facility. It's a living building. Right now, BGI is sequencing 4,000 genomes a week, all fully automated, no human intervention. And this is largely a computer science facility because someone has to analyze all that data. So along the top, these are all dormitories for visiting scientists to help them analyze the data. And this is crazy. They've got their own aquaculture, their own agriculture, so they can be self-sufficient. And this, I toured it. This is actually built now. On the left, they just broke ground for that. That's about a $5 billion U.S. facility. And they've got dorms for their staff on the left. Yeah, it's solar powered. It's again, the center area here, this whole area is vertical agriculture, it's aquaculture. so that they can feed their staff and genetically engineer foods and experiment with them. So this I mean, it sounds funny, but you know, China has a lot of pollution. They're aware of that. They have a huge population, and so they can identify, for example, fish that have very high protein, that grow fast, and they can make them larger. Right. So it's very interesting, and again, 4,000 genomes a day. Right now they've done 1 million cell-free DNAs. They do it to identify aneuploidy in pregnant women. And they've identified 6 women with cancer, subclinical cancer, right? So this is the opportunity this place presents. And this is the MOU we signed. We won't go into detail, but it greatly expands our relationship, ultimately ending up in the possibility of Vancouver's Shenzhen Institute, because BGI is based there. So, and sequencers and technology development, so on and so forth. I thought, you know, people ask me what I'm doing over there all the time. This is, you know, Guanxi means relationship. And so this is sort of an idea. So we're sequencing roughly 100 tumors for Dr. Black. We've got 65 Chinese tumors, prostate, with matched normal genomes, transcriptomes, deep RNA, and microRNAs. We have a manuscript in preparation on that. We're supporting a circular RNA project, which could be amazing blood-based biomarkers. We've got one of YZ Wang's xenograph models, which is, you know, this was probably close to $200,000 in sequencing they covered, and this led to a signature for metastasis and intermediate and lower grade tumors. A manuscript is up ready to submit on that. It supported the CPC gene project out of Toronto, sequenced 175 prostate tumor transcriptomes, tumor dormancy, so on and so forth. And the ones with asterisks have all been paid for in China, either collaborators in Shanghai or BGI. So you can add this up, it ends up being quite a lot of money. So the reason I'm here is to talk about, you know, work coming out of the core. It's going to be helped a lot by BGI. And so we all know about this concept of precision oncology. Most people tell you it's very simple. We do every omic analysis we can think of, and then we target the tumor and cure the patient. The problem with this is it really boils down to predict and pray, right? Because we don't really know. You know, the mutations have to be in the proper context. You know, we don't even know what combinatorial therapy is. Not really. So it really comes down to predict and pray. And most people are aware in the field that it doesn't really work that often. And now I was thinking about this, and you know, prostate cancer is actually a perfect example of how difficult this really is. So globally, there's 900,000 diagnoses a year, 250,000 deaths, and this is despite having one of the best biomarkers for any cancer, second, third generation targeted and even combinatorial therapies that hit the drivers, dihydrotestosterone and the androgen receptor, and in the background of good chemotherapies. And we're still not curing the disease, right? We've hit every major buzzword in genomic medicine or precision medicine or personalized, whatever your favorite term is, right? And we're still not curing the disease. And so this is a slide that illustrates what many here are well aware of now, and that is when we treat adenocarcinoma with these new potent drugs, we can get at least a bifurcation and possibly a trifurcation. So roughly 75% of the time you get reactivation of the AR, and we get CRPC. About 20 to 30% of the time we get neuroendocrine transdifferentiation, so no longer expressing AR, expressing neuronal markers. And then at some frequency, there's these double negatives that Pete Nelson describes. They're AR minus, chromogranin minus. And so a couple of years ago, I reviewed a paper on cell-free DNA that I didn't believe. It was, you know, it looked too good to be true. And so I asked Sas Volek in the group to replicate the data, thinking I was going to have to write to the editor because we had to accept the paper. We couldn't find anything wrong. And so that's led now to 3 papers. And the concept of cell-free DNA is that lets you look in the blood for circulating tumor DNA. And so to date, studies of therapeutic resistance and metastatic CRPC and other cancers have been constrained by spatial and temporal heterogeneity. And so I call this Genomics or dynamic genomics. And the idea is that these minimally invasive liquid biopsies can overcome spatial and temporal heterogeneity, can reveal the dynamics of resistance and guide precision oncology and drug development. Again, the people really driving this are Stas Volek, who designed all the initial experiments, then Alex Wyatt, who took it and ran with it, and then of course Kim Chi, who's brought the patients to us. And so the concept is shown here, and this is cell-free DNA is distinct from circulating tumor cells, which there's not many of in prostate cancer. So on the top, you can see the circulating tumor cells. That's not what we're talking about. We're talking about the circulating tumor DNA, which exists in a background of circulating normal DNA, right? Crapone, if you will. And we have to sequence deeply through the normal to get to the small amount of tumor that's there. And so the circulating tumor DNA provides a representation of tumor heterogeneity because theoretically you're seeing it from all metastatic sites. It's easy sampling because it's non-invasive. You get larger amounts, so you get like 2.9 nanograms per mil of blood. That's roughly 500 genome equivalents compared to 6 picograms from one circulating tumor cell. It has a half-life of roughly 3 hours, so we're getting a real-time readout. And we can detect down to 3 to 10% of total cell-free DNA, maybe. That number is always moving up. Some companies and some techniques can get down to 0.1% now, but that's tough. So the first time we did this was a very simple hypothesis. We had a mixed cohort of heavily treated guys. And the hypothesis was simply that AR gene aberrations will be detectable in cell-free DNA and will associate resistance with agents targeting AR in the signaling pathway. And we had 62 guys progressing on abiraterone, enzalutamide. And we did whole genome analysis using array CGH and targeted sequencing of the androgen receptor ligand binding domain, part of it. And this was all possible because Stefan Lebian and my group convinced me to talk Roche out of one of their sequencers and give it to us for free. And it worked, surprisingly. So this was done with 454 technology initially. So first thing is Ann Hager in the lab ran array CGH on all the guys for whom we got enough cell-free DNA to do array CGH. And so from chromosome 1 to 21, we're looking at copy number changes, so red are deletions and blue are amplifications. And, to me, as a genomics guy who studied prostate cancer for a long time, this looks like prostate cancer. So that was good, and then we compared it to 181 primary tumors published by Taylor et al. And you can see that overall they look pretty similar. And then the genes that stand out, RB, AP, AQ, many of these are diagnostic for prostate cancer. So this gave me confidence that it was working. And then we could look in more closely at individual chromosomes and look, for example, at MYC amplification. And this was working with the AACR-PCF Stand Up to Cancer West Coast Dream Team. So we have matched biopsies and circulating tumor DNA. So on the top left, you can see a focal amplification of MYC. On the right, from the biopsy, and on the right from the cell-free DNA. So with cyclin D and cyclin E stands out as being different. This could be due to heterogeneity and adaptation or whatever. I just reviewed a paper from another group, and they see basically exactly the same thing. Small numbers, but the same phenomenon. Disturbingly, they see focal amplifications driving resistance in only 5 weeks on treatment. sad, probably a sad reality that we're learning. So this is, again, the ability just to look at copy number by array CGH, looking at cell-free DNA in the top, and we're looking at androgen receptor amplification and biopsies on the bottom. So the take-home message here is that there's concordance between the biopsies and the cell-free DNA, which you know, this really gave me confidence we weren't dealing with some kind of a sexy artifact. And then we were able to sequence a small region ligand binding domain. And this is quite interesting and disturbing because we did this, and some of these patients were able to do multiple time points. So if you look, this is time point 1, that this guy has 3 haplotypes, so maybe 3 metastatic tumors leaking DNA or actively secreting it, as some people think. And time point 2, he has, I think that's 5 haplotypes. There's another guy who went from 2 haplotypes to 6 haplotypes. So it presents a very complex picture of the disease. And this was from the Clinical Cancer Research paper. This just shows, this is a messy way to look at it, but this just shows that in general, there's a mutual exclusivity between amplification of the androgen receptor and mutations of the androgen receptor. So you get one or the other most of the time. You don't get both. And then in a paper just online now, in JAMA Oncology, this is really led by Alex Wyatt and Kim Chi, the more sequencing of the AR and ligand binding domain, looking at pre-treatment, on-treatment, and post-treatment here. And what you can see between the pre-treatment and the post-treatment is sort of dynamic remodeling of the genome in response to therapy. And that on therapy, it's quite difficult to detect cell-free DNA, presumably because therapy is working, and it's much more difficult to see. And we can also look at this sort of association of AR gene status at baseline with progression-free survival on enzalutamide, and this simply shows that, again, we have copy number changes up here, mutations here. And if you have aberrations, either mutation or amplification in the AR, guys do worse than if they don't. So look at the left and the right, and Kaplan-Meier show, of course, that these AR aberrations, guys do worse as RB, which is not surprising at all. And then again, this shows this is a busy slide. I won't go through it in detail. But this just shows the sort of dynamic remodeling that goes on sort of at progression. So again, there's copy number changes on the top, mutations here, and other events here detected using BGI instruments they gave us. And all these arrows and dots signify changes that happened between baseline and progression. So again, very dynamic situation once these guys go on therapy. And again, this is in line with the paper I just reviewed, where they see huge remodeling at the level of genome amplification, focal amplification, many of the genes for which there are targeted therapies for, they claim. So again, this is a real testament to the prostate center. We just work together, right? And so this guy, Artem Cherkasov, who most of you know, just came to me and said, hey, look, you've got mutations. I've got 3D protein work and in vitro systems. Let's functionalize all these mutations. and I guess in like 6 months, we had a paper ready. So, you know, he was able to map all these different mutations into the AR and have some ideas of what they do, develop an in vitro system to get a readout on what the mutations are doing. And this is the result of that. So this is a patient who went from two haplotypes to 6 haplotypes, okay? And you can look up here, and when he modeled how were these mutations working, we found that the first guy, patient 12 here, was progressing on bicalutamide. And in his in vitro functional assay, he found that bicalutamide was now antagonizing, was agonizing those mutations. So it was acting like gasoline on fire. I know those mutations are roughly 50-50 in the circulating tumor DNA. So this guy was switched to enzalutamide, and those mutations were 50-50, dropped to
both in today?
I think that these two here, so like 15 and 5%. And they're being killed by enzalutamide, but now all these other mutations are starting to be agonized by enzalutamide. So this is actually, you can read this now in Genome Biology. So again, this points to very dynamic remodeling on treatment, and is consistent with our previous work and other papers coming out right now. And this is one of Martin's patients who, again, you know, the treatment history on the top left, biopsies, mutation mapped by Artem Cherkasov in the 3D structure, and found again that his mutation was being agonized by bicalutamide, was predicted to be antagonized by enzalutamide, and I also found out that the mutation was causing the dihydrotestosterone to become a superagonist, right? Not just a normal role, but superagonist. So this is normal activity here and with mutation here. And so this guy was switched on to abiraterone and enzalutamide and had a very positive outcome. So we can study dynamic genomics another way, right? And that is working with my brother Y.Z. Wang and his extraordinary postdoc Don Lin. That is using these patient-derived xenografts for precision oncology and developing insights to mechanisms of resistance. And so many of you have probably heard this story where he can graft human prostate tumors into the mouse on the kidney capsule, and in this case, he put an adenocarcinoma on the kidney capsule, called it 331, waited 4 to 6 months, and the adenocarcinoma turned into neuroendocrine cancer. And you can see that because it goes from being AR positive and PSA positive, AR negative, PSA negative, and then positive for synaptoseason. And the patient had the same outcome. So these models are, I call, high fidelity, right, in terms of genomics, gene expression, immunohistochemistry, and many times accurately modeling the patient outcome. This just shows that in the patient, the adenocarcinoma ultimately, I think 6 or 7 years later, turned into neuroendocrine cancer. So we looked at, we looked at the, in the PDX model, we looked at this. So that is a great model for looking at clonal evolution, right? So we analyzed the genome structure on the left, that's array CGH, again, that's just looking at copy number. You can just think of it as barcoding. We did deep sequencing and identified the number of fusions, for example, Hempersburg, and it turned out that they were exactly the same at all loci. We've now done deep exome sequencing, same mutation spectrum. So this is not clonal selection. This is a state change. So the adenocarcinoma cells turned into another kind of cell. So it's really a phase transition or state change. And so because it's a PDX model, we were able to do a time point study. And so this was work done by Don Lin and Alex Wyatt in the lab. And so we just did a series of time points as it transitioned from adenocarcinoma to neuroendocrine. And you can see the immunohistochemistry here, the AR dropping off, PSA dropping off, and then here the emergence of neuronal markers, right? So no AR, no PSA, but the neuronal markers at the end once it terminally differentiates. And because we can do the time course, we can actually ask what's happening. right? And over time, what genes are going off, what genes are coming on. And so this, what I'm showing you here is we can see the AR genes being turned off as we move toward neuroendocrine. You get the transiently expressed genes that come up, little, go high, and go off again. And then persistent genes come up and stay on. And so you can look here and see these are at androgen receptor signaling axis genes. Watch this gene rest. We'll talk about that more than when signaling apoptosis, cell fate, and cell growth here, and neuroendocrine markers. And so these come up, right? Lately stay on, so on and so forth. So we get this dynamic view of gene expression. And so the interesting thing is we had sequenced a few neuroendocrine tumors when I first got here, sort of by mistake. And when we sent the paper in, we were told, this is interesting. Actually, one of the reviewers said he was disappointed because it wasn't a very good paper, because we hadn't analyzed all the mutations, and it was reviewed by genomics people. What we did find, however, was biology. And so we were able to see that RAS was a significant player in neuroendocrine cancer, and we were able to identify splicing factors and other elements that looked very interesting from the point of view of biology. Right. And so we identified this gene as being alternative. RAS is being very active. And so we pursued this. And it's important now that I tell you that RAS is a master regulator of neurogenesis. Right? So its job is to make sure that neuronal genes are never expressed in non-neuronal tissues, because it programs things like cell-cell communication, axon growth, all things that you might imagine are bad for non-neuronal tissues. And it gets spliced. It gets a neuronal specific intron spliced in, which leads to this truncated form that can no longer bind and turn off genes. Okay, so it normally turns off roughly 1,900 genes in the genome in non-neuronal tissues. And when it gets spliced, it can't do that anymore. That's all you need to remember. So I was in Tianjin, China, talking about this at a urology meeting, and I ran into this guy, Amilcar Flores Morales, who was giving basically the same talk, and Steve Ball from Harvard, who was giving the same talk. So we decided to collaborate, and this is work that I did with Amilcar Flores. And so, I'm not going to go into all the details here, but what he found with us and with our models was when you have androgen receptor pathway inhibition, you de-repress, you cause de-repression of this molecule beta CRP, which is an ubiquitin. That then allows RAS to get ubiquitinated and degraded by protease substrates. So remember, I told you that REST keeps neuronal genes turned off in non-neuronal tissues. So when you have androgen pathway inhibition, right, the REST protein starts to get degraded. You start to get expression of neuronal genes in non-neuronal tissue. And we knocked it out. We showed, yeah, this is all happening. If you do it in LinCap, they don't look neuronal, but we start getting expression of all the neuronal markers, synapses, and so on and so forth. So we concluded that loss of REST is necessary for the neuronal phenotype to establish, but that it's not sufficient for transdifferentiation. And so again, the great thing about working at the Prostate Center is I'm an okay cell biologist, but Shushen Dong is a great molecular biologist, and YZ is a great biologist. And I have a big group working in computer science. So we're able to really dig into this neuroendocrine transdifferentiation issue and ask what's going on. And we were able to identify, just now, it's online at European Urology, show that the splicing factor, SRRM4, drives neuroendocrine transdifferentiation. And so I'm just showing some of the computer science over in the top left where Nilgan Donmez in my lab is able to get an incredible splicing index to separate. So normally you would use gene expression to separate adenocarcinoma from neuroendocrine. She used exon splicing and got a much better result. And this just shows that this is work from Xu Shen's lab that in the context of loss of RB and P53 and androgen pathway inhibition, that SRM4 can not only induce in 2D culture a neuronal phenotype in WNCAP cells, but can drive it to form 3D spheroids that are neuroendocrine. And in addition, he's shown that can get neuroendocrine xenografts for the first time this way. So here's the working model of what we think is going on. So SRM4 is a context-dependent driver of neuroendocrine transdifferentiation. So androgen pathway inhibition leads to rest protein degradation, right? And that can set up a neuronal phenotype, NEP. Now, SRM4 comes on late. Okay, we don't know what turns that on yet, although we think it's probably epigenetic. That reprograms the mRNA. So now you've got loss of protein and loss of mRNA, right? So you get the strong neuronal phenotype, and then in the presence of P53 or RB loss, then you get neuroendocrine transdifferentiation. So we think that P53, RB, Aurora kinase, and these other things are facilitators, but that REST and SRM4 are the master regulators of this process because they are in neurogenesis, and what we're getting is a neuroendocrine phenotype. But it's absolutely dependent on androgen pathway inhibition. So another gene we found using the 331 model is PEG10. And again, it was these three guys. Shisuka Akamatsu is like a brilliant molecular biologist and surgeon, really dug into this. And this just shows that I won't go into all the gory details here, but PEG10 comes from the lower left panel, a giant bubble, comes up very strongly immediately post-castration, right? And we show that real-time PCR comes up huge post-castration, and again, at the terminal differentiation in the neuroendocrine type. And then we show that as well by immunohistochemistry. So the gene expression holds at sort of microarray transcriptome level, and RT-PCR, quantitative RT-PCR, immunohistochemistry. And then Alicia Beltran was kind enough to show that in her cohort of neuroendocrine that yeah, PEG10 is highly expressed in neuroendocrine tumors as well. And so, you know, what is this gene and what does it do? This is a topic for like 2 seminars. This gene is so complicated. So I'm going to give you sort of high-level overview. So it's a retrotransposon derived gene, so it's like a viral gene, right? It's only 120 million years old. And we know this because it's required for placental development. It's maternally imprinted, so there's a competition between the male genome and the female genome to control expression of this gene. It's got an active -1 ribosomal frame shift, which is incredibly rare. It's got an active protease domain like HIV, and it's a very limited expression in adult tissues. The gene model is shown over there. You can see that it has 2 start sites, 2 stop sites, and a ribosomal frame shift. And it's got that protease domain. So it can make this sort of repertoire of splice variants and protein isoforms that have this very complex expression pattern over the course of going from adenocarcinoma to neuroendocrine. And so poor Xu, he thought he was working on one gene, he ended up working on 3 genes in essence. And what came out of all this was this idea that in adenocarcinoma, you have high AR, and that keeps PEG10 turned off. Because remember, we kill AR by either castration or enzalutamide. PEG10 comes up screaming tenfold right away, right? But in neuroendocrine, when there's no AR, PEG10 is expressed. One isoform drives invasion. Another isoform drives proliferation. And another isoform that we did not publish on, but someone else did, protects against apoptosis under stress. So I mean, this is needed for placental development, right? So invasion, proliferation, apoptosis, and you might speculate that other isoforms are modulating the immune system. So from this comes this model, sort of putting it all together. So as I was thinking about this last night, PEG10 seems to come in here to protect stress response to keep the cells alive. And then later, once we have terminal differentiation, remember PEG10 goes way up again at terminal differentiation. It's driving proliferation, invasion, and I would bet modulating the immune system. So I think this is a decent model for what's going on in androgen pathway inhibition-induced neuroendocrine transdifferentiation. This is obviously just a roadmap, and there's a lot to do. So YZ and I have been thinking about this concept, I think, since I got here. Advancing from predict and pray to evidence-based precision oncology, and because of a confluence of events, I think we're here. Because of BGI, donors, and the large CFI grant, I think, and other grants that are submitted, we can finally think seriously about going from the patient biopsy or cell-free DNA to omics, to panomics, to network analysis, which, because of Faraz and Schenk, are very good at, to predict drugs, to test them in the mice for evidence-based precision oncology, to treatment, sort of hopefully virtuous circle that will benefit patients, and in a very integrated way with, prostate center, the clinic, BGI. This really ought to be possible now. And everybody has their anecdotal evidence that this works. Ours is this ovarian patient. She had ovarian serous adenocarcinoma. We profiled the tumor and put the tumor in mice. The profiling suggested that Avastin and Herceptin would work. This was supported in the mouse model. So Herceptin, Avastin, and then control were just fine. So what was the outcome of this? This note, from her husband, saying that I think she had been 6 years since diagnosis and 5 years since recurrence. So this is the kind of thing that gives one confidence that, that this is actually, if you can find targets and you can get the tumor to grow. But as I told her husband, I said, this is a Hail Mary. If it works, it's because you got lucky, not because we were good. And I think that's the truth. That's the truth. We could have found all kinds of mutations that predicted all kinds of drugs, or for which there were no drugs, right? Like RAS. What are you going to do? And then I'm going to end with this slide because I think this is a really, scientists. I always tell my students that if you talk to a rock and ask the right questions, it will talk back to you. It will tell you how old it is, how it was formed, where it was formed, on and on, right? So Howard Sewell, Prostate Cancer Foundation, talked academician soon into paying for a lot of sequencing on one of YZ's models. So YZ has this model where he took 5 biopsies and made 5 xenograft lines. These all have different metastatic potential, from no metastasis to strongly metastatic within a certain time frame. as he would say, all things are conditional. So we did deep genome sequencing. We did deep transcriptome sequencing. I think we did microRNA sequencing, right? The hypothesis being we will find a mutation that's linked to metastasis or gene expression, right? Academician assumes also a general in the military. Chop, chop, guys, find me something. I paid a lot of money for this. We found nothing, zero. General's not happy, right? So, and that makes me nervous being in China. So we found nothing. I mean, it was ridiculous. So then Fan Mo, a postdoc in my lab, who I met in Hangzhou, China, and Dong Lin started thinking, well, what's different between them? You know, maybe it's the stroma, right? And this became a really tractable problem because in the PDX models, the human stroma gets replaced by mouse stroma. Okay, so now we can just bin the genes. If they're human genes, they're coming from the tumor. If they're mouse genes, they're coming from the stroma. So they're telling me they're doing this, right? I'm like, okay, whatever. But this is interesting, have fun. So then it started to look more like they might be finding something. So we struck up a collaboration with Genome DX. And we looked in the Mayo Clinic at the signature they derived that they said would predict metastasis. So in the Mayo Clinic, we got this result, right? And then, so that's interesting. So then we looked at Gleason grade 7, we got this result. And so I just presented this data to Dana-Farber, I think 2 weeks ago, and Massimo Loda got all excited. It's the same data, but from patients. So this is amazing, right? But by any measure, just looking at the tumor, there's no difference. So this is about to be submitted. We may do, depending on how far along Massimo is, we may do a joint submission. But and then we're looking at lower Gleason grade as well. So clearly, if this is true, it means it's the stroma that's driving metastasis, not tumor genes. And there's no reason to think this isn't true. So to conclude, I think that despite far too much hype, tremendous progress is being made toward delivering on the promise of genomics. It's not quite like the days when Francis Colin said, if we sequence, it will cure human disease. That was 2001, I think. We're not there yet, but we're getting there. And delivery will require an integration of genomics, computer science, biology, and medicine, but it will also require regulatory agencies and healthcare systems to adapt to a very different clinical landscape. And increasingly, I think this is international, right? Because many things are going to be developed in China, and they can test scalability in ways we can't. And they have to force down healthcare costs. Because if you think the Canadian healthcare system is out of control cost-wise, think about 1.7 billion people, right? And in the near future, this will lead to greatly improved disease management and increased quality of life rather than cures, because evolution is a doggedly powerful force. And as I showed you, you perturb these systems, and they come back to bite you in unexpected ways. So with that, I will finish and thank my large cohort of collaborators. And I tried to mention everybody as I go through my talk, so if I forgot anybody, I'm sorry. Thank you.